I'm Dr. Maria Cristina De Las Llagas, and welcome to part 1 of the preview of cell biology. For this part, we are going to look briefly at the origin of cell biology as a discipline. Then we will consider the three main historical strands of cytology, genetics, and biochemistry that have formed our current understanding of what cells are and how they actually work. Later on in part 2, we will conclude with a brief discussion of the nature of knowledge itself by considering biological facts, the scientific method, and the use of common model organisms in cell biology, so that we would be able to answer the question of how do we know what we know right now. The cell is the basic unit of biology. What exactly does this mean? It actually tells us that every organism either consists of cells or in itself is a single cell. Therefore, it is only by understanding the structure and the function of cells that we can appreciate both the capabilities and the limitations of living organisms, regardless of whether they are animals, plants, fungi, or microorganisms. And we are now in the midst of an exciting trend in cell biology studies, practically because we have technology that are made available for us so that we can better understand how cells work. Therefore, we could actually say, because of this technology, cells are constantly changing. The previous idea that cells are static, as can be seen in text shown in illustrations, is actually a misconception. Cells are capable of growing, they reproduce, they become specialized, they have an ability to respond to stimuli, and they are also capable of adapting to changes. All of these characteristics and properties of cells point out that cells are constantly changing and the nature of cells really is dynamic. The field of cell biology, or otherwise we call as cytology, in itself is rapidly changing because several scientists are able to work together in order to gain a better understanding of how cells work. For example, the convergence of cytology, genetics, and biochemistry has made modern cell biology one of the most exciting and dynamic disciplines of biology. The story of cell biology started to unfold more than 300 years ago as European scientists began to focus their crude microscopes on a variety of biological material, ranging from tree bark to bacteria to human sperm. One such scientist was Robert Hooke. He was the curator of instruments for the Royal Society of London. In 1665, Hooke was able to build a microscope, and he examined thin slices of cork cut using a penknife. What he saw was a network of tiny box-like compartments that reminded him of a honeycomb, and he called these little compartments cells. We know now that what Hooke observed were actually not the cells at all. He was only able to see the cell walls of a dead plant tissue. But he did not understand that they are actually dead because he did not know that cork cells could be alive. While it is true that he was able to notice that the cells in other plant tissues were filled with fluid, which is simply called as juices, Hooke preferred to concentrate on the more prominent cell walls of dead cork cells that he had first encountered. So Hooke's observations were actually limited by the magnification power of his microscope. When we say magnification, it means the ability of his microscope to enlarge objects, which was only 30 times their normal size. and so it was difficult to learn more about the internal organization of cells. A few years later, Anton van Leeuwenhoek, a Dutch merchant, was able to produce small lenses that could magnify objects 300 times their size. And using these superior lenses, Leeuwenhoek was able to become the first to observe living cells, such as blood cells, sperm cells, bacteria, and the single-celled organisms, which we know now as algae and protozoan found in pond water. Two factors, however, restricted further understanding of the nature of cells. First, the microscopes of the day had limited resolution or resolving power. This is the ability to see fine details of a structure. In fact, even the superior lenses that was used by Leeuwenhoek could indeed magnify an object 300 times. However, the resolving power or its resolution is not that good. In other words, he was not able to see the intriguing architectural details that are actually found inside the cells. More than a century has passed before a combination of improved microscopes were actually developed. Of course, another thing that limited the study of cell biology would be the descriptive nature of cell biology itself. Most of the scientists during that time focused on what they were only able to observe. They had limited emphasis or focus on the reason why the structure is such. By 1830s, an important development happened, which was the development of the compound microscope. By this time, there are now two lenses in the microscope. One lens, which is the eyepiece, magnifies the image created by the second lens, which is the objective lenses. This allowed both higher magnification as well as better resolution. Now, structures only 1 micrometer in size could be seen clearly using compound microscopes. Aided by such improved lenses, the English botanist Robert Brown found that every plant cell he looked at contained a rounded structure. He called this dark, rounded structure as nucleus. In 1838, his German colleague Matthias Schleiden came to the important conclusion that all plant tissues are actually composed of cells, and that an embryonic plant will always arise from a single cell. A year later, a German cytologist by the name of Theodor Schwann reported similar conclusion concerning the animal tissue, thereby discrediting earlier speculations that plants and animals do not resemble each other structurally. These speculations actually arise because plant cell walls form a conspicuous boundaries between cells, and these are readily available even using a very crude microscope, in that these cell walls are actually absent from animal cells. As originally postulated, the cell theory had two basic principles. In 1839, Schwann postulated the cell theory such that, first, all organisms are consist of one or more cells. Second, that the cell is the basic unit of structure for all organisms. Less than 20 years later, a third principle was added, This grew out of Brown's original description of the nuclei, and it was extended by the Swiss botanist Carl Nageli to include observations on the nature of cell division, such that by 1855, Rudolf Virchow, a German physiologist, concluded that cells arouse only by the division of other pre-existing cells. Therefore, Virchow encapsulated this conclusion in the now famous Latin phrase omnis cellula e cellula, which can be translated as the third principle of the modern theory of the modern cell theory, which says that all cells arise only from pre-existing cells. This tells us, therefore, that the cell is not only the basic unit of structure for all organisms, but that it is also the basic unit of reproduction. No wonder we really need to understand cells and their properties so that we could appreciate all other aspects of biology. This figure shows to us a few examples of the diversity of cells that exist in our world. So that cells, we know, exist in a wide variety of shape and sizes, from the filamentous fungal cells, to the spiral treponema bacteria, to the spherical biconcave disc human red blood cells, down to the diatoms, and even to the retinal neurons. All of these are examples of diversity of cells that exist in our world. Modern cell biology results from the weaving together of three different strands of biological inquiry: cytology, biochemistry, and genetics. By cytology, it focuses mainly on the cellular structure and emphasizes the optical techniques that are used or needed in order to observe the details and structures of a cell. The biochemistry part, the second strand, represents the contributions to our understanding of cellular structure and function. Most of the developments in this field have occurred over the past 75 years, though the roots could go back at least a century earlier. Especially important in biochemistry is the development of techniques such as ultracentrifugation, chromatography, radioactive labeling, electrophoresis, and even mass spectrometry that are necessary or essential for separating and identifying cellular components. The third strand contributing to the development of modern cell biology is genetics. Here, the timeline for genetics stretches back more than 150 years to Australian monk and biologist Gregor Mendel. Again, much of the knowledge that we know of genetics has occurred within the past 75 years. An especially important landmark on the genetic strand is the demonstration that in all organisms, the DNA, or the deoxyribonucleic acid, is the bearer of genetic information. So here is a figure that shows to you the timeline of the development of modern cell biology. Strictly speaking, cytology is the study of cells. Historically, however, cytology has dealt primarily with cellular structure, mainly through the use of optical techniques. In a little while, we will describe briefly some of the microscopy that is important in cell biology. Meanwhile, a very important challenge of understanding cellular structure and organization is complicated by the problem of size. Most cells and their organelles are too small to be seen by the unaided eye. In addition, the units used to measure them are unfamiliar to many students and therefore are often difficult to appreciate. The problem can actually be approached in two ways. By realizing that only two units are really necessary to express the dimensions of most structures of interest to us, and by considering a variety of structures that can be appropriately measured with each of these two units. And what are these units? I'm talking about the micrometer, otherwise also known as the micron. This is the most useful unit for expressing the size of cells and organelles. A micrometer is 1/1,000,000th of a meter. In general, bacterial cells are a few micrometers in diameter, and the cells of plants and animals are 10 to 20 times larger in any single dimensions. Meanwhile, organelles, such as mitochondria and the chloroplast, tend to have diameters or length of a few micrometers and are thus comparable in size to whole bacterial cells. In general, if you can see it with a light microscope, you can express its dimension conveniently in micrometers. Another important unit of measurement is the nanometer. This is the unit of choice for molecules and subcellular structures that are too small to be seen using a light microscope. This particular figure will show to you that you describe a plant cell, a chloroplast, and even mitochondria and the animal cells in terms of micrometers. However, for the world of the nanometer, in other words, those that are very, very small, we could describe them in terms of nanometers. For example, those that are too small to be seen with the light microscope. A nanometer is 1/100000th of a meter. A ribosome has a diameter of about 25 nanometers. Other structures that can be measured conveniently in nanometers are the diameter of the microtubules, the microfilaments, which is 7 nanometers, and even the DNA double helix, which is only 2 nanometers. A slightly smaller unit, which is the angstrom, is occasionally used in cell biology when measuring dimensions within proteins and DNA molecules. An angstrom is equal to 0.1 nanometer, which is about the size of a hydrogen atom. The light microscope was the earliest tool of cytologists, and it continues to play an important role in understanding cellular structures. Because of light microscope, we are able to see and identify membrane-bound structures such as the nuclei, the mitochondria, and chloroplast within a variety of cell types. These structures are what we call as the organelles. These are prominent in plant and animals but are not seen in bacterial cells. Another important advancement in observing cells includes the development of the microtome sometime in mid-1800s, as well as the availability of dyes and stains during that time. A microtome is an instrument that is developed for rapid and efficient preparation of very thin tissue slices of biological samples. Meanwhile, dyes are important for staining and identifying subcellular structures. So far, in visualizing cells, we make use of white light. That's the reason why light microscopy is also called bright field microscopy, because white light is passed directly through a specimen. And of course, a specimen would be more visible if it is stained as compared to unstained. This figure shows the relative resolving power of the human eye, the light microscope, and the electron microscope. Notice that the vertical axis is on a logarithmic scale to accommodate the wide range of sizes that are actually shown there. As used in microscopy, the limit of resolution refers to how far apart adjacent objects must be in order to be distinguished as separate entities. For example, if the limit of resolution of a microscope is 400 nanometers, this means that objects must be at least 400 nanometers apart to be recognizable as separate entities. In other words, the smaller the limit of resolution, the greater is the resolving power or the ability to see fine details of a structure using a microscope. There are a variety of specialized light microscopes that have been developed. There is phase contrast microscopy, differential interference contrast microscopy, fluorescence microscopy, and confocal microscopy. In the case of fluorescence microscopy, it allows the detection of proteins, DNA sequences, or molecules that have been made fluorescent by binding to antibodies. An antibody is a protein that binds a particular target molecule called an antigen. Therefore, in fluorescence microscopy, the antibody will bind to a target molecule, which is the antigen, making it highly specific therefore. An example of a stain that is used would be the green fluorescent protein. It can be used to study the temporal and spatial distribution of proteins in a living cell. In the meantime, confocal microscopy uses lasers and special optics in order to eliminate a beam on a single plane within a specimen, meaning only those regions within a narrow depth of focus are imaged or can be seen, and regions above and below the selected plane of view will appear black or it will appear blurry. When it comes to digital video microscopy, it uses video cameras in order to collect digital images. So here is a diagram showing the different types of light microscopy and its comparison. A major breakthrough in resolving power came with the development of the electron microscope, which was invented in Germany in 1931 by Max Noll and Ernst Ruska. This is so significant because light microscopy in itself is subjected to the limit of resolution imposed by the wavelength of light used in order to view the sample. In the case of electron microscope, it uses a beam of electrons that is deflected and focused by an electromagnetic field. Because the wavelength of electrons is so much shorter than the wavelength of photons of visible light, it only means that the limit of resolution for electron microscope is much better than that of the light microscope. In fact, it can resolve as little as 0.1 to 0.2 nanometers. Furthermore, as a result, the useful magnification of electron microscope is also even higher. It can magnify up to 100 times. Most electron microscopes are of two types: the transmission electron microscope and the scanning electron microscope. The transmission electron microscope uses electrons that are transmitted through the specimen, while the scanning electron microscope allows one to visualize the surface of a specimen as it is being scanned by detecting electrons that have been deflected from its surface. These are examples of images that can be seen in details using the electron microscope. Figure 1 shows a breast cancer cell. Figure 2 shows details of the sunflower pollen grains using a transmission electron microscope. Figure C shows a cat intestinal epithelial cells, and Figure D shows a mitochondrion taken from bat pancreatic cells. The biochemical strand studies the chemistry of biological structure and function. At about the same time, cytologists started exploring cellular structure using their microscopes. Other scientists were making observations that began to explain and clarify cellular functions. Much of what we now call as biochemistry dates from discovery reported in 1828 by a German chemist by the name of Friedrich Wöhler. Friedrich Wöhler was a German chemist, a contemporary and fellow countryman of Schleiden and Schwann. He was able to revolutionize our thinking about biology and chemistry by demonstrating that urea, which is an organic compound of biological origin, could be synthesized in the laboratory using inorganic starting material, which is ammonium cyanate. Prior to the work of Wöhler, it has been widely held that living organisms were unique. They are not governed by the laws of chemistry and physics that apply to the non-living world. By showing that a compound made by living organism could be synthesized in the laboratory just like any other chemical, Wöhler helped to break down the conceptual distinction between living and non-living worlds, and he was able to dispel the notion that biochemical processes were somehow exempt from the laws of chemistry and physics. Another major advance came about 40 years later when French chemist and biologist Louis Pasteur showed that living yeast cells were responsible for the fermentation of sugar into alcohol. In 1897, the German bacteriologist Eduard and Hans Buchner found that fermentation could also take place with isolated extracts from yeast cells. That is, the intact cells themselves were actually not needed in order for fermentation to happen. Gradually, it became clear that the active agent in the extracts were specific biological catalysts, which we now know as the enzymes. Significant progress in our understanding of cellular function came in the 1920s and 1930s as individual steps in the biochemical pathways for fermentation and other related cellular processes were elucidated. You're probably familiar with the Embden-Meyerhof pathway, and then of course the Krebs cycle. All of that are actually biochemistry. Crucial in understanding of cell biology would be biochemistry methods. For example, subcellular fractionation. This method uses centrifugation in order to separate or isolate different structures and macromolecules. Another important biochemistry method would be chromatography. This is a technique that is used to separate molecules from a solution based on size, charge, and chemical affinity. Electrophoresis, on the other hand, uses an electrical field to move proteins, DNA, or RNA molecules through a medium based on size charge. This is something that we also do in our laboratory. Meanwhile, after proteins have been separated by electrophoresis, Mass spectrometry is another common method that can be used to determine the size and the composition of the individual proteins. This technique allows researchers to determine the identity and characteristics of individual proteins in a mix of thousands of different proteins in a cell. And because of this led to an emerging field in biology, which is proteomics, where researchers attempt to understand the functions and interactions of all proteins that are present in a particular cell. This is an image showing a chromatograph of plant pigments and an image showing proteins that have been resolved or separated through electrophoresis. The third strand in the historical cord of cell biology is genetics. Just like the other two, this strand has important roots in 19th century. In this case, the strand begins with Gregor Mendel, whose studies with the pea plant he grew in a monastery garden must surely rank among the most famous experiments in all of biology. His findings were published in 1866, laying out the principles of segregation and independent assortment of hereditary factors. Now we know this as the genes. In the decade following Mendel's work, the role of the nucleus in the genetic continuity of cells came to be better appreciated, such that in 1880, the German biologist Walther Flemming identified structures that are darkly colored inside the nucleus as the chromosomes. Flemming called the division process as mitosis, from the Greek word for thread, because the chromosomes resemble thread-like structures. The chromosome number soon came to be recognized as a distinctive characteristic of a species and was shown to remain constant from generation to generation. That the chromosomes themselves might be the actual bearers of genetic information was suggested by German anatomist Wilhelm Roh as early as 1883 and expressed more formally by his countryman biologist August Weismann shortly thereafter. With the roles of the nucleus and the chromosomes established and better appreciated, the stage is now ready or set for the rediscovery of Mendel's initial observations. This came sometime in 1900s, when the study of Mendel were cited almost simultaneously by three plant geneticists working independently. These were Karl Correns in Germany, Ernst von Zermatt in Austria, and Hugo de Vries in Holland. Within 3 years, the chromosome theory of heredity was formulated, following work by American physician Walter Sutton and German biologist Theodor Boveri. So the chromosome theory of heredity proposed that the hereditary factors responsible for Mendelian inheritance are actually located on the chromosomes within the nucleus. This hypothesis received its strongest confirmation from the work of American biologist Thomas Hunt Morgan and his student Calvin Bridges and Alfred Sturtevant at the Columbia University during the first 2 decades of the 20th century. In their studies, they made use of Drosophila melanogaster as their experimental model organisms. More advancement on the understanding of the DNA was made, for example, The first isolated DNA was called nuclein by Frederick Miescher, and it was known to be a component of chromosomes by 1914. Experiments with bacteria and viruses in the 1940s began to implicate the DNA as the genetic material. American biologists George Biddle and Edward Tatum, working in 1940s with the bread mold Neurospora crassa, formulated the one gene and one enzyme concept, asserting that each gene controls the production of a single specific protein. Shortly thereafter, in 1953, an unlikely team of former ornithology student James Watson and physicist Francis Crick, with assistance from X-ray crystallographer Rosalind Franklin, proposed the now famous double helix model for DNA structure, which immediately suggested how replication and genetic mutations could happen. Later on, it was discovered how DNA specifies the order of amino acids in proteins, and that several different kinds of RNA molecules serve as intermediates in protein synthesis. The three important kinds of RNA molecules that are needed for protein synthesis or translation are the messenger RNAs, the ribosomal RNAs, and the transfer RNAs. While it is true that we follow the central dogma, there are exemptions to the central dogma which would include viruses that uses the RNA genome. For these viruses, they make use of the reverse transcriptase, which is an enzyme that allows viral RNA to synthesize a complementary DNA molecule. So this is a diagram that shows the general flow of genetic information from transcription to mRNA transport and eventually to protein synthesis, which is translation. In working with DNA, recent technology, such as recombinant DNA technology, makes use of restriction enzymes. These enzymes would cut the DNA at specific sites or places, allowing now the scientists to combine recombinant DNA molecules with DNA coming from a different source. Another technique, called DNA cloning, is the generation of many copies of a specific DNA sequence. Meanwhile, when you say DNA transformation, it is when you introduce DNA into a cell. From here, the challenge of analyzing the vast amount of data that can be generated from DNA sequencing has led to a new discipline which is called bioinformatics. This merges computer science and biology as a means of making sense of the sequence data that was obtained. This approach has led to the recognition that the human genome contains approximately around 25,000 protein coding genes, about half of which were not characterized before genome sequencing. It is because of this that scientists are now able to study the proteome. Proteome is the total protein content of a cell, and analyzing thousands of proteins simultaneously in what you call as proteomics. So proteomic studies aims to understand the structure and properties of every protein produced by a genome, as well as to learn how these proteins are able to interact with each other in order to regulate the function of the cells. There are a lot of bioinformatic tools that are publicly available through the National Center of Biotechnology Information, and these are just some of the activities that you will perform firsthand in your laboratory. With technology, we are now able to branch out the field of omics into different sub-branches, such as transcriptomics, which specializes on the study of all genes transcribed in a cell, metabolomics that analyzes all metabolic reactions happening at a given time in a cell, lipidomics, the study of all the lipids in a cell, ionomics, the study of all the ions in a cell, and all the other possible omics that will emerge in the future. So how do we know what we know now? What we think of as facts today are things that are known to be true because of what is available to us right now. In other words, technology allows us to understand what we now understand. Ideas that are replaced earlier facts could now be considered as misconceptions because of unavailability rather of technology in the past. This is the reason why we say that biological facts may turn out to be incorrect. What we know right now are subject to change in the future because knowledge or information gained through science is actually dynamic. And thus, there is a need for scientists or researchers to search for significant scientific literature using peer-reviewed sources or journals so that they would be able to come up with a good hypothesis and that they could test this hypothesis using the correct model. In cytology, cell cultures are commonly used as model systems. These are used to study cancer, viruses, proteins, and cellular differentiation. Although we have advanced microscopes and even sophisticated methods to isolate and analyze individual cells and cellular components, there are still some data that we cannot understand unless we are able to study it using intact organisms. Thus, cell biologies also rely heavily on a variety of model organisms representing the major kingdoms of life to help unlock the secrets of a cell. A model organism is a species that is widely studied and well characterized, easy to manipulate, and has particular advantages, making it useful for experimental studies. A few examples are the bacterium Escherichia coli, the yeast Saccharomyces cerevisiae, the fruit fly Drosophila melanogaster are just some of the model organisms that we know. So this is a diagram showing the different model organisms that are commonly used in some research or experiments. These model organisms are very crucial because well-designed experiments are needed so that the results of your study or experiment will be considered valid.